to watch the
He
video.
was sick. He called me this morning. He's like,
To that,
yeah.
somebody on the camera.
So we're actually starting the brain. And by the time we're done with lecture tomorrow, we will be done with both components of the prosencephalon. So remember, I said we're going to keep going back over this material, and you'll hear it multiple times. So remember the three primary vesicles in the development of the neural tube, and the brain component of that were prosencephalon, mesencephalon, and brachiocephalon. Okay? The prosencephalon then divided into telencephalon and diencephalon. So today we're going to talk about telencephalon, and we've already covered part of the telencephalon when we described the lateral ventricles. So the lateral ventricles are components of the telencephalon. Okay, we've already covered that part of the ventricular system. Today we're going to talk about the lobes. So we're going to start off with the occipital lobe, the temporal lobe, the parietal lobe, and the frontal lobe. Parietal lobe grows, the temporal lobe grows really fast, and also covers another structure of cortical surface tissue known as the insula. So all of these structures you need to be able to recognize as belonging to the telencephalon. Within each of them, we're going to talk about certain components: the hippocampus, the precentral gyrus, the postcentral gyrus. And so recognize that any of their components as well are going to be a part of the telencephalon. Okay. Then tomorrow we're going to do diencephalon, which is thalamus and hypothalamus. And then starting on Tuesday, we're going to work on the mesencephalon and brachiocephalon structures. Okay. Tuesday is also the day that we do our brain modeling. So you'll need the 4 colors of clay. If all you have is Play-Doh, that works, but not as well. It tends to lose its shape and it doesn't hold up very well. And so if you can get the 4-color modeling clay, you can get it at Target or Walmart or Michael's from anywhere from $1 to $3. And if you totally forget on Tuesday, I've asked the bookstore as well to stock it. So I've turned it in with my book orders. So they should have it there. All right. So when we look at the brain surface area, we immediately notice that there are gyri. There are folds and valleys. The folds are called the gyri, and the valleys, by folds I mean like a hill-like structure, and the valleys are the sulci. First time I saw a human brain, well, I saw a movie. I don't know what my parents were thinking, but when I was 5 or 6, they took myself and my sisters, who were older and younger, to some movie about a drunken thing you would see for driver's ed, about a kid that was drinking alcohol and got in a crash and was killed, and the coroner did the autopsy. The medical examiner was slicing his brain, and I remember that vividly. And then when I was in high school, I went to high school in Maryland, just outside of Washington, D.C., and we took a field trip with my biology class to Georgetown University Medical School. And I remember 2 things from that trip. One, that they desperately needed bodies donated to science for cadavers, and they were paying people $1,500 to sign a contract that when they died, Georgetown University could have their body. So I remember joking with my friends that, well, who knows whether they're going to come in the middle of the night and take your body before you're ready to give it up. The other thing I remember was opening this door, and it was a relatively small room. It wasn't a classroom size, maybe 15 by 15. And there was this, what I thought was an older gentleman who was in his 42, something like that, 40 or 42. He'd been an engineer, and I don't know why, if he got tired or was always wanting to go to medical school or whatever reason, but he quit being an engineer, and he was a freshman medical student, and he was studying the brain. He was sitting there all by himself in this room studying the brain. And he was memorizing the names of every single fold and every gyrus, and every sulcus, every valley. And I just remember when I started to take neuro thinking that this is what I was going to have to do, was memorize every single one of these. And we did learn about 10. You're going to learn about 3 or 4, so not much difference. But there's so much new material in medical sciences, from the chemistry and the pharmacology and the cell biology. They still go to school for just 4 years. So they have to cut back on some things and leave that for the specialists. So if you were going to go in to specialize in neurology, then obviously you would have to learn all of those. So when we talk about a gyrus and a sulcus, you've probably seen pictures, or if you scuba dive, seen actual brain coral. And that's because it looks like the surface of the brain. So if we look at a part of the brain here, this is the medial surface of part of the brain. And so each fold that I'm creating here is going to be a fold of gray matter. So this area right here is a gyrus. Okay? This area right here is a gyrus. So the gyrus is like the hill. Gyri with an I instead of a US is plural. And the valley is the sulcus. And they all have names. This long space, in fact, if you guys want to take down some of the brains and have them with you while we're going through this material, that's probably a good idea. This space here that we talked about containing the falx cerebri is the longitudinal fissure. So typically a fissure is supposed to be like a deep sulcus. But there are times when it seems they didn't follow that rule and they used the term fissure when they should have used sulcus and vice versa. So on this picture that I have up on the board, this is 2 cerebral hemispheres. The cerebral hemisphere is the frontal, parietal, temporal, occipital lobes of one half of the brain. And this gap that you could put a piece of paper down, where that falx cerebri is the longitudinal fissure. Okay? So that's the deep fissure between 2 halves of the brain. And then we have the central sulcus. And the central sulcus is a shallow groove. We're going to look at a sagittal, a lateral view, a side view of the brain. In this view here, not quite halfway. As humans, we have a lot of thinking capability. So we have a larger frontal lobe, say, than a sheep. And so back here, about just slightly past halfway, not quite 2/3, we have a fold that is passing down the lateral surface. It actually starts on the medial surface about 3/4 of an inch and follows around on the lateral surface. And this is known as the central sulcus. And it's going to separate the frontal lobe from the parietal lobe. Okay? Then you can see here, and this actually goes by a little bit longer, we have a lateral sulcus. That's where the C-shaped growth of the brain brings the temporal lobe forward and outward and forward. So this is the lateral sulcus. And it separates frontal lobe from temporal lobe. You should recognize these terms from the bones that are in contact with these surfaces. So this would be the front of our brain. Okay. So we have the central sulcus, longitudinal fissure, and it's a little difficult here to see the lateral, but let me change the view of that. So here you can see the lateral sulcus, and here is the central sulcus. Okay, so a little bit past halfway. Now on the models, those can be hard to find. Well, the lateral sulcus isn't, but the central sulcus is because it doesn't follow on a real human brain. You can actually see on the medial surface where this starts, and then follow it back. So it kind of starts here and then curves over, and you can find it on the medial surface and then turn over. But go about 2/3 of the way back. So on lab exams, I actually use a model that has that drawn in. So I'm not showing 3 different ones and you have to choose which one it is, okay? So that's the view from the lateral surface of the brain. And then if we turn it over and look on the medial surface, so remember this is the surface that's supplied by the anterior cerebral artery. So the medial surface, this is the cerebellum, so this is the back of the brain stem, thalamus and hypothalamus. Here would be, this is where the 3rd ventricle is. We open up this slit here, we would be in the lateral ventricle. Here's the cerebral aqueduct and the 4th ventricle, all right? So here's the frontal lobe. Here would be see how this comes in right here? That's the central sulcus right there. And then if we come around, this deep groove right here is the parietal occipital sulcus or parietal occipital fissure. And that separates parietal lobe from occipital lobe. So you can't see it on the other side, but you can see it from this medial surface. And you can see it on your models as well. So if I turn the brain around and flip it, you would have this is kind of what we're seeing here. This would be the front of the brain. And we have a gyrus here. And then we have, at this angle, we have the parietal occipital sulcus or fissure. All right? And that's going to separate the parietal lobe from the occipital lobe. This one is one of those that's in between. It is deep, so sometimes it's called a fissure, but it doesn't completely separate the lobes from each other, so sometimes it's called a sulcus. And so this would be the parietal lobes and occipital and frontal. And from this view, except for a little bit down here, we're not seeing the temporal lobe. You can see the temporal lobe down there. Okay.
So it's parietal occipital fissure.
This would be the left, yes. You can see the same thing on the right, but you're looking at the left, and this would be the same model that you're holding. Yeah. Okay. Now, we also have gyri that are named. So these are the grooves, these are the valleys, but we also have gyri that are named. So here's our central sulcus. Just in front of the central sulcus is the precentral gyrus, meaning the gyrus in front of the central sulcus. So and then there's another one behind it that belongs to the parietal lobe. So we'll talk about the function, what goes on in these gyri, when we talk about the frontal lobe. But this is the precentral gyrus. So it's a fold of gray matter like we see here that runs laterally down the side of each hemisphere. one in front, that's the precentral, and one posterior to the central sulcus. So this is the postcentral gyrus. Tomorrow we're going to talk about the limbic system, and we're going to talk about the cingulate gyrus. So we have to look at the medial view. So before I move to that, here is our central sulcus running through here. So this gyrus is the precentral gyrus, and the posterior is our postcentral gyrus running through here. Okay. And then if we turn to the medial view, this is the corpus callosum. We're going to talk about that in just a couple of minutes. And this is the cingulate gyrus right here. Okay. It's involved in emotions, it's involved in fear, it's involved in making decisions because of fear, motivation due to fear. So there's a variety of, we'll talk about that tomorrow. But that's the cingulate gyrus. And down here in the temporal lobe, so if we look at this view, wrong way, we look at, okay, where's my upside down brain? Upside down brain here. So turn your brains over, so you're looking at the inferior view. Notice how the temporal lobe expands almost as much on the inferior surface as you saw on the lateral surface. So this is still all temporal lobe. It's going to rest on the greater wing of the sphenoid bone. And this inner portion right here is the dentate gyrus. So we'll bring that up again when we talk about the temporal lobe today. Okay? So just on the inside, so here's the temporal lobe, and along here is the dentate gyrus. So on the medial surface of the temporal lobe, here would be the dentate gyrus. Okay? So those are the 4 gyri that you need to identify. The precentral, postcentral, cingulate, and dentate. And cingulate is missing from that list. I mean, we'll talk about it tomorrow, but if you want to add it to all the gyri here in the same place, you can add it to the list of gyri on page 52. Under dentate, you can add cingulate. C-I-N-G-U-L-A-T-E. Remember that pattern of C shape that I talked about with the lateral ventricles? We're going to see that over and over. We see it with the cingulate gyrus, has that C-shaped pattern. The anterior cerebral artery that's lies in cingulate gyrus travels over it in that C-shaped pattern as well. All right, now let's look at white matter. Remember what the gray matter was when we talked about the
Unmyelinated.
That's for unmyelinated tissue, which is primarily cell bodies. And white matter is myelinated tissue, the nerve fibers. All right? So when we look at various sections, and we'll start to do this tomorrow, of the brain once we've covered the internal content, we have 3 types of white matter that are available. Association, commissural, and projection fibers. Okay? So this diagram of the red fibers that you see are the association fibers, and this diagram was determined by actually scraping away brain tissue. So it was really a startling lab the first time I did this at Davis. We used the brains that were dissected during gross anatomy in the fall, were kept in the spring when we took or taught, and we both, neuroanatomy. The students were given a breadboard essentially with one of the brains and a tongue blade. And they literally used the edge of the tongue blade to scrape away the surface of the brain and go through gray matter to white matter to gray matter as we kind of went from superficial to deep. And you end up with structures that look like this. All right, so the first type of association fibers are short association fibers. Short association fibers are going to connect information from one gyrus to another gyrus in the same lobe. all right? So this gray matter is going to send a short nerve fiber to this cell body in the next lobe. And that is a short association fiber.
I mean, gyrus.
Did I say gyrus?
You said lobe.
In the same lobe. So short association is gyrus to gyrus in the same lobe. For an example, when we look at the brain, the gray matter in the precentral gyrus, I talked about right here, is formed by the cell bodies of neurons that are going to go down to my spinal cord, synapse with another cell body, and go out to a motor neuron. So when I decide to extend my fingers, right about here on the side of my brain, a bunch of cell bodies are firing and going down to the spinal cord. But I can do that as a conscious thought. I am going to point my finger. So I consciously know that I need to extend my elbow, extend my wrist, and point my finger. All right? However, if I'm going to walk, I don't think consciously about, unless I need to extend, flex my knee, dorsiflex, point my toes up and down. I just walk. I don't think about each movement that I need to do. If I'm playing the piano, I don't think about each finger that I need to move unless I'm just learning the song. If I haven't memorized, I just play, or any other instrument. So just outside the precentral gyrus in this area, and we'll talk about it towards the end of lecture when we talk about the frontal lobe, are several gyri known as the premotor cortex. That's where that planning is already determined. So for things that are repeated movements, my brain already has known, because it's done this over and over, which of the muscles in here it needs to activate in what sequence. And so it would activate them using short association fibers, coming from the premotor area to the precentral gyrus, and activate those muscles in sequence. Okay? So those are short association fibers. Another example of a short association fiber is in the occipital lobe. We have a primary cortical area, primary visual area. And again, we're going to go over this when we get to the occipital lobe. I'm just give you a couple of examples. What this happens is as an electrical current from my retina passes via neurons back to that occipital lobe, an image is created. So I see white, I see red, I see gray. That's what is happening in the primary visual cortex. But then I know what I'm looking at. I know that it's a table. I know that it's a quiz sheet or even a piece of paper. I know that it's the wall. So that requires short association fibers linking from the primary visual area out to those where I've already laid down memories of things that I've seen before. Okay? So it's associating. I know the meaning. I know the object that I'm seeing. It's not just there. All right? I can see that. You know, sometimes you look at a piece of paper, but you know that if you look long enough, you can kind of see through it and see three-dimensional structures. So that involves the association area. Just seeing that black and white pattern is your primary. But then as you start to kind of see through it, and you start to see a shape to something, that's kind of how the association area works. We associate known things that we've seen before. And that's still gyrus to gyrus within the same lobe. Okay? Well, then we have long association fibers. So let's say that I see something and I recognize, that I've seen it before, but now when I see that, I remember a word. I know that that is called the hood. And I know that that hood is to use to take out noxious fumes. All right? Knowing all of that involves memory, that involves association with the bottom of my frontal lobe. That's a long association fiber. So it's going from gyrus to gyrus, in different lobes, but in the same hemisphere, on the left side of the brain or on the right side of the brain. It doesn't cross over from one side to another. So a long association fiber is gyrus to gyrus from different lobes. In the same hemisphere. When I was in high school, for some reason, my sisters and I decided to throw a dinner party for my parents. My older sister made a paper mache statue of a woman holding a bottle of water that was about 3 feet high. And went all out. Made, spaghetti and dessert and all that kind of stuff. And we had a tiny little room, probably 10 by 11, that we called the music room. It was just big enough for the piano and the TV in one chair. And so we moved the chair out, set up a little card table with a tablecloth and everything. Well, the day that it came for the set up a printed invitation, the day that it came for the dinner, the 3 of us girls all came down with the flu. But we still fixed the dinner and served it to our parents and laid on the floor while they ate it, sick as dogs. So it took about a year before I could smell tomato sauce and not feel nauseated because I associated it with that dinner, okay? Remember, association. So that smell, which is temporal lobe, the long association fiber to my frontal lobe. So that's what we mean by gyrus to gyrus, different lobes, the same side of the brain. All right, so short association, long association. And so here would be short association fibers, and this would be long association fibers. Then we connect gyrus to gyrus in different lobes on opposite sides of the brain. There are 3 major types of commensura fibers, but we're just going to learn one, and that is the corpus callosum. So we've seen that already. It forms a band. Let's say I want to form an association fiber from this gyrus to that gyrus. All right, so it crosses from the same lobe, say the temporal lobe on this side of the brain, to the, well, this is really frontal lobe, to the frontal lobe on that side. Or from, so this view is a transverse view, top of the head taken off, looking down on the brain. So this would be frontal lobe, occipital lobe, and temporal and parietal, okay? This is a coronal section. So this is cutting the brain like it was a slice of a loaf of bread. And it could be either the frontal lobe or the parietal lobe. Based on what I see here for the nuclei, we're just at the transition from frontal to parietal. All right, we are just up here at the area of the lateral ventricles where we're transitioning between posterior horn and inferior horn. And we don't have the thalamus yet. And so here we see the corpus callosum going from the same gyrus on one side to the same gyrus on the other. But more importantly, that is, or more specifically, that is illustrated by this view. So frontal to frontal, occipital to occipital, temporal and parietal to temporal and parietal. So I've drawn fibers like that on here, so frontal and posterior. They actually bend under each other. So the front of the corpus callosum and the back of the corpus callosum are thicker than in between because of that folding under. So when we look at a view of the corpus callosum from our human brain here, this is the corpus callosum right here. Underneath this is a thin membrane that we have to cut to see into the lateral ventricle. Here is the thalamus. So it looks like the fibers are traveling in this direction. They're not. They're coming out at you. Because remember, they're coming from this side of the brain, going through here, and going to the other side of the brain. So they're actually cut in cross-section. They're not traveling front to back. They're traveling around here, side to side here, and around here like that. Okay? So the best view to help you recognize that is this one here. Fibers are going from, so for commissural fibers, the fibers are going from gyrus to gyrus in the same lobe but opposite sides of the brain.
That's in different hemispheres, correct?
Correct. Gyrus to gyrus, same lobes, opposite hemispheres. Now this creates some interesting situations when the corpus callosum is cut. So I remember watching a video when I was in college about this young man. It used to be the treatment for severe epilepsy back in the early 30s and 40s, but it was abused, like electrical shock treatment, and in the last 15 years or so it started to come back into use again with specific individuals. And the treatment is to cut the corpus callosum, so cut through these fibers. And so the information is still coming in to the right side of the brain or the left side of the brain, but isn't being shared. All right, so what I feel with my right hand crosses over and is being processed in the left side of my brain. When I decide to move my right hand, that originates from the left side of my brain. So this individual was seated before a screen, and he could only see the screen with his right eye or his left eye. So he was shown various objects, say an orange with his right eye. Now, he could say the word orange, okay? Well, actually, if he was right-handed, he wouldn't be able to say the word orange because the information would be processed for speech on the left side of his brain. If he was left-handed and had speech on this side, he could say the word orange. Then he was asked, but he could see it was orange, and his brain would know it was orange. But he couldn't say the word orange if his speech center was on that side. If you asked him to reach into a paper sack and pull out what he saw on the screen without telling him what it was, he wouldn't be able to do that. Because he was seeing the orange with his right side, but his right hand was processing that information on the left side of his brain, and that wasn't being shared. So he could tell you, I'm touching an apple, I'm touching an orange, I'm touching a baseball, but when you said, okay, which of those, pick out which of those is on the screen, this side of the brain didn't know that, because this side of the brain saw the screen, this side of the brain was processing his sensory input. And that's similar to the little story here. So a split brain patient sits in front of a screen, words were flashed on either side. When a word was flashed on the left side, they couldn't say what they had seen because their speech center is on the left side and that was being processed to their right, and they couldn't share that information back and forth. So it's a very similar experience to what I saw on the video. So this is the corpus callosum. We've looked down through the longitudinal fissure, and the part of the right hemisphere has been scraped away so that you're seeing more of these fibers. And here, you're just kind of looking down. So if you, in our sheep brain, you'll be able to see this. Pull apart the longitudinal fissure of the sheep brain and look down inside, and you'll see that corpus callosum. We'll have to cut through it to actually open up the sheep brain and see both halves. Okay. This is an individual child who was born without a corpus callosum, and it actually can cause some developmental problems and some problems with the limbic system development. Remember I talked about the cingulate gyrus, so we'll look at those problems tomorrow.
Is that what happens to somebody who has no visual memory? I know someone whose daughter has no visual memory, so she sees things like for the first time always.
Every time.
Like, yeah, she doesn't know
That
that.
could be the visual association area. It could be part of a problem with the hippocampus for laying down short-term memory, but if it's just associated with vision, it sounds like it's part of the problem with the association areas. Because if it was corpus callosum, if she saw it on one side, yeah, she'd be able to see that. If she can't remember it all, it sounds like it's more association area. All right, then our third type are projection fibers. These are fibers that are sensory or motor and project up or down. So We're going to be looking at these in more detail on Friday, Thursday, and Friday of next week. No, Thursday, Friday, and Tuesday of next week. Last Friday of next week and the following Tuesday. But anyway, the last 2 days before our exam, we'll be looking at these in detail. So I'm going to draw a smaller one of these, the coronal section of the brain. This would be the lateral sulcus I'm drawing in here. Lateral ventricle and third ventricle. Brain stem. The spinal cord. All right. So when I touch this tabletop, I'm activating receptors for pain, touch. Well, hopefully not pain. For touch. And that's being sent in on a pseudo-unipolar neuron into my spinal cord, which activates another neuron. It's going to cross over, and it is going to synapse in my thalamus, which we'll talk about tomorrow. And then another neuron is going to end up in my post-central gyrus. All right? So this would be my parietal lobe. And my post-central gyrus. These two fibers right here are projection fibers. Okay, they're projecting upward towards my post-central gyrus for processing of that pain, touch, temperature sensation. Now in response, I decide to pick up my hand and look at it. Okay? So that requires a motor. Now we don't usually have split brains like this, but just so I don't have to draw it all over again, this is going to be the frontal lobe on this side. They're really about 1.5 inches or so apart. And so I'm going to move my fingers and my elbow. So that requires activation of a neuron. Oops, wrong side, sorry. Well, I'm going to do it on this side anyway because I put the other floater. So it requires activation of a neuron. I would usually move the same side of my body, but because the motor pathways on that side, I have to draw it like that. And so that's a projection neuron. It's traveling down, you remember, going down the spinal cord. So these are projection fibers, projecting up or projecting down, brain to brainstem, spinal cord, brainstem to brain. So when we look at our sensory and motor integrative pathways Friday next week, we're going to be talking about integrative fibers. And that's what's shown here. All these fibers that are passing up and down between the spinal cord and brain are projection fibers.
Can you repeat exactly what projection fibers getting sense of vast velocity?
Okay, projection fibers project upward or downward within the central nervous system. So a sensory fiber going from spinal cord up to thalamus or thalamus to the post-central gyrus, that's sensory going towards the central nervous system, is a projection fiber. In response, the motor fiber going from the precentral gyrus down to brainstem or spinal cord is also a projection. So they don't go forwards and backwards or side to side within the brain, they go up and down.
Okay.
Okay? So motor and sensory pathways here of how we send motor information down to the spinal cord or receive sensory information up from the spinal cord.
The motor goes straight down,
It's going to cross over.
but how the sensory goes to thalamus, it skips.
Yeah, the upper motor neuron goes all the way down to the spinal cord. There's only 2 neurons involved. And we'll look at that next week. But yes, you caught on to that. All right, so the commissural fibers on this diagram are showing the corpus callosum here. These fibers that are going in an up and down direction are the projection fibers. now we're ready to look at the actual lobes of the brain. And you will see a pattern as we move from this week into next week between motor and sensory, that posterior or dorsal structures of the spinal cord and brain are sensory, and anterior or ventral, and those terms are interchangeable, tend to be motor. Yes, Sam?
I was just gonna ask, assuming we're moving on, external white capsule?
Okay, so that's the corona radiata are these fibers right here. Most of the projection fibers make this internal white capsule. It's just a name for all these sensory and motor fibers together that are traveling up and down. The external white capsule is out here outside the basal nuclei, which we'll talk about tomorrow. But internal white capsule or corona radiata, you can see the term here, are these fibers passing between the thalamus and what we'll talk about tomorrow, the basal nuclei, and external white capsule is out here. But most of them are in the internal. So we're going to start. Our motor responses are responses. They're a motor action because of a sensory stimuli. So I hear something, I feel something, and I say something or I move something in response to that. All right. So we're going to start with occipital lobe. And here the various lobes and their components are shown in color. So I'm going to repeat this slide over and over with each of the lobes. And the reason we know this information is because the brain can be conscious while it is having surgery. It does not have pain receptors. Okay, it does have neurons. I told you about Dr. Carson's comment tonight, this summer. Ben Carson is a famous neurologist. He got into political hot water because he's a conservative, and he spoke at a prayer breakfast for the president, and they railed on the Obamacare. And he's a neurosurgeon from Johns Hopkins. And this summer, my kids and their class were back east in Maryland, and he's a member of the church that we went to while we were back there, the entire class. And so the kids were they'd had to read a book of his for class, for one of their English classes as freshman or sophomore. And they'd heard ahead of time that he was a member of this church. And they were so psyched. We sat right behind him. So all these 20 high school kids are taking pictures of the back of his head. And with their cell phones and sending them, I saw Dr. Carson. So he gave up to give a little homily, a little talk about the body and things that you could learn about it. And he said, and there are no neurons in the brain. And my mouth just dropped open. And I turned to my husband and I said, wrong, 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 wrong. And so the history teacher who was leading it, I spoke to him after. I said, look, I didn't mean any disrespect to Dr. Carson. Obviously, he knows that there are neurons in the brain. Good grief, he operates on these. What he meant to say was there are no pain receptors in the neuron. There are neurons that receive signals and process it as pain. But when you cut the nerve tissue itself, unless you're cutting those particular neurons that process pain, I could cut the occipital lobe and it wouldn't hurt. Okay? I could cut the frontal lobe and it wouldn't hurt. The only place it might hurt is parts of the postcentral gyrus where I actually process pain. And so when individuals need to have part of the brain removed for tumors, they are often conscious during that procedure. They have local anesthetic for the skull and the scalp and the meninges, because those obviously have pain receptors, but the brain itself doesn't, and these people are kept conscious so that they can use small, tiny electrical currents and stimulate various parts around where that tumor is or whatever it is that they want to remove, and make sure that they're not removing something they don't want to take out. And so for the last, 70 years or so of doing this, we've mapped out, scientists and physicians have mapped out where different functions are. So if I stimulate this part of the brain, my knee is going to contract or flex. If I stimulate this part of the brain, my lips are going to move. If I do it on this side, I'm going to feel a tingling or pain sensation in my lips. And so they have numbers. And as you look at your lecture notes, for the various parts of each lobe, I've indicated what these numbers are. They're called Brodmann's numbers. You don't have to memorize them. I don't test you on them, but certain maps of the brain that you see will have those numbers there. If you take a psychology class, they may talk about this, a behavioral psychology class, and so I've just put those numbers there as a reference, but they're not something that we have to identify. But that's how we know what these parts of the brain do, okay? As well as having people who have damage to that area and a loss of that function. So here you can see the parietal occipital sulcus, and this whole pink, orange, whatever area is the occipital lobe. So that's the first area that we're going to look at, this area right here. Okay. And you'll notice that it has regions, so the very tip of it, if you were to just kind of pinch the very tip of the occipital lobe, it is identified, that would be what's covered up here. It's this bright yellow area here. That is the primary visual area, or the primary visual cortex. That's what I mean when I say if you were to stimulate that with an electrical current, you are going to see various colors of light or a sensation of something moving. You probably won't see an image per se, but that's what happens when we see something. An electrical current is carried back through the thalamus to this primary visual area or cortex. Images are produced. A blind man was hooked up to electrodes into this area, and the electrodes were governed by a computer. You know how you can take books, put them on top of certain computers that will read the pages in a kind of electrical mechanical voice? Well, this same similar type of a computer, instead of reading it, would send electrical currents to these electrodes in this man's brain, and he would see spots of light in a braille pattern. So the computer would convert the words, the letters of the words, to the braille pattern of raised dots, but instead of raised dots, they were lights. And so he quote unquote saw in his brain, it's the back of his retina, these patterns of lights, and he read the patterns of lights like he could feel the patterns with his fingertip. Because stimulating those cells with electrical current created a light image. So that's the primary visual cortex. It's signaled by objects moving across your vision, by various colors and so on, all right? And then the association area, which is the area around it, so this more mustard color area is the visual association area. So if I were to see this piece of paper, I would just see it. My visual, primary visual area just would indicate that there's something white with black on it. It doesn't tell me it's a piece of paper. It doesn't read the words for me. It just says there's an image there. Okay? Now that visual association area with short association fibers is going to connect those words and that piece of paper with things that I've seen before. The first time you show a piece of paper to a baby, it doesn't know it's a piece of paper. Okay? It just sees it. And then it learns that what paper is and what it feels like and what it does. So even when I see that paper, I can imagine in my mind what its texture is, all right? When I look at the words, now long association fibers are going to connect with memory on the bottom of my frontal lobe, and I can read that word, not just see it, but I know that that's an A I'm looking at, and I know that the A and the R and the E make the word R, and I know what that word means and how it's used in a sentence. So that requires long association fibers. But the short association fibers go from primary visual cortex to visual association area and allow me to associate that as a 3D object, okay, and what that structure is to provide some meaning to it. So that's why I think that your friend's daughter is having problems with that area rather than the corpus callosum itself. All right, so then if somebody is blind, it would mean that either their retina is not working, well, you could say their cornea maybe, or their lens isn't working, or their retina, or the optic nerve, or the thalamus, or the primary visual cortex. All of those are places where damage could result in blindness. If they can actually see it, but they can't use it, then we're talking about association problems. Okay? And those are probably the most frustrating for their friends and family, because it's hard to understand how someone can see something and yet not recognize it for what it is. Jane Goodall is a behavioral animal scientist studying chimpanzees in Africa, and she has an association area where she doesn't recognize faces. So she sees somebody, she knows it's a body, it's a face, but there's no meaning attached to that. She could say that's an eye and that's a nose, but she doesn't see any difference from one person to the next. And so she can't recognize somebody that she's met before. I don't know if she could even say it's a female or a male. Okay, I'm not sure how. And that involves part of the parietal lobe as well, but primarily is a visual association problem. Did you have a comment? Okay. All right. So there are whole sections of the primary visual cortex. Some measure color, some are fired by movement. If something moves across your vision, lateral to right or up or down, so it talks about motion here. Obviously, you don't have to know each of those sections, but the brain is very organized, okay, and how this is how we are able to understand what we've seen, and brain learns this. Okay, temporal lobe. So temporal lobe is separated from parietal and frontal by the lateral sulcus, which extends, kind of peters out here, extends slightly past the central sulcus. It's involved primarily in two aspects: hearing and short-term memory. So for hearing, just on this posterior edge of the lateral sulcus, we have our primary auditory area, or cortex. Again, this is just hearing a sound. So when I make a sound, you hear that. All right. Now, when you heard it, some of you might recognize that as a metal sound, okay, or a slap, or my voice. But you heard the sound. That involves the middle ear and inner ear. That's all working, and it's being processed as a high note or a low note in that primary auditory area. Then short association from primary to the auditory association area is going to allow you to attach meaning to that. Was it your child's voice? Was it a bird call? Was it a trumpet versus an oboe playing the note, B flat? So all of those associations are learned as you develop and involve short association fibers to that auditory association. Then again, long-term memory association with other components. So when I was in college, I took a humanities course. This is before CDs were used. And so my music teacher, humanities teacher, playing songs from various areas, Renaissance. Every time he played a particular song on this record, the needle skipped. And on our final exam, he played the same song and the same needle skip, and I didn't have to hear anything else. I immediately knew what song it was. And most of you have heard you only need 2 or 3 notes of a song. There's TV games made out of this, and the rest of the song starts playing in your head. All right? So that involves connection with the basal part of the frontal lobe for long-term memory to bring up those components. Then we have the hippocampus. So this is, if you turn your brain upside down, remember I talked about the dentate gyrus? All right, where this is kind of rolled up into the inferior horn. So this gap right here with choroid plexus, this is the lateral ventricle, and it's curving around. It's curving around this way and then forward as the inferior horn. So that would be this area right here. Here's the inferior horn, and underneath that is the hippocampus that forms the floor of the inferior horn. This is part of what our play modeling on Tuesday will help you with, is visualizing this, all right? And the hippocampus is coiled. You can see how it's kind of coiled, and it reminded somebody of a seahorse's tail. That's what a hippocampus is a seahorse. And so this is where short-term memories start. There's a communication between the hippocampus and the mammillary bodies of the hypothalamus. So here's thalamus. This would be the third ventricle right here. Tomorrow we'll look at the thalamus and hippocampus, hypothalamus. And neurons go between these two as short-term memory is laid down. And so when the hippocampus is damaged, you can't lay down those first memories. If you don't create short-term memories, you can't make them into long-term memories. So this was an area that with Alzheimer's is being studied. They started noticing changes in the neurons and the presence of plaques because loss of memory, short-term memory, eventually long-term memory as well is associated with Alzheimer's. Back in the 50s, I believe it was 50s, Saylor liked to make models, and his roommate would tease him. Well, when he was holding the two pieces, they didn't have fast-drying acrylic glue, and he had to hold the pieces of the ship or the plane together, and so his roommate would take advantage of this to poke at him. And he couldn't do anything about it because he had to either drop his model or keep holding it. I have no idea why the guy was using an ice pick, but he would poke him with this, and the guy had enough, and he whirled around really hard, and the ice pick went into his brain, okay, into his hippocampus. Now, it only did one hippocampus, but that was enough. The other one, you're not typically cited with memory, but anyway, that was enough damage that he lost his short-term memory. And people would go to visit him, and it was the typical, hi, how are you, come in and sit down, and then 5 minutes later, he'd introduce himself and act like he'd never seen you. So when he died, the medical field had talked to him, and he had agreed to donate his brain. And as they sectioned through it, the hippocampus was atrophied on that area. It was one of the early cases where they started making association between the role of the hippocampus and memory function. So that's what I want you to recognize the hippocampus for. So obviously we'll see it as part of the telencephalon because it's part of the temporal lobe, but associate it with memory or lack thereof. All right, then in addition to that, well, let's do the parietal lobe first, and then we'll come back to the sensory speech area because it's part of the parietal lobe. So let's move to parietal lobe now. And that's this area right here. So it's kind of the most vague as far as what's separating it. So on the medial surface, we have the parietal occipital sulcus or fissure. On this surface, there's no really good groove that separates parietal lobe from occipital lobe. And even our lateral sulcus kind of peters out. So there's no real nice definitive separation, but essentially this area here is the parietal lobe. It's certainly separated from the frontal lobe by the central sulcus. Right? And it has the major gyrus that we talked about earlier as the post central gyrus. So that is shown here in darker blue as the post central gyrus. So the drawing I'm going to make on the board is going to be a cut through that post central gyrus. All right? Similar to what I had up here already. So here's the longitudinal fissure. I'm drawing the medial surface. All of this is in the post central gyrus. Here's the lateral sulcus. And then we have the temporal lobe. And here would be the post central gyrus on the other side. Have our lateral ventricle. And at this section, back here, by the time we come through here, we're in the third ventricle. Our thalamus, part of the brain stem, and spinal cord again. All right. So remember, I'm just drawing what I drew over here earlier, just a little bit larger. So if I feel something on my lip, what nerve, cranial nerve is that? Trigeminal, okay? That's going to come into the brainstem. And another neuron will take that up to the thalamus. And that's going to come into my gray matter of my post-central gyrus. If I feel something from my knee, that's going to come in and be processed in this area. So along the post-central gyrus, which is made up of the gray matter of all these cells that are processing and actually creating the sensation of pain or touch or temperature, because if it doesn't go to this neuron, I'm not going to feel touch. Okay? So if I cut it here, I'm not going to have any sensation of touch. It has to reach that to be processed. So that I have specific regions of the body then that are matched to specific areas. And you can see this on the bottom of page 64. This mapping again came from clients that were conscious during brain surgery. And so the knee is right here at the, what is the weird looking ring? Is right up here at the bend. Down the medial surface is where the leg and foot are processed. Notice the size. So there's a lot of neurons that are involved in the lip. So the bigger the structure of the body, the more neurons are devoted to processing sensations from that region. All right. Notice the size of the jaw and the teeth compared to the lips. It barely is existing there, and yet the teeth can be really painful when they hurt. But as far as the number of neurons involved in processing that. Okay. The term homunculus comes from man. So this is just kind of the shape of a man or individual stretched out with the number of neurons reflecting the size of the structure. So this is the processing of pain, touch, temperature, light touch, pressure sensations from, this would be general somatic afferent, coming into the post-central gyrus. So if I had a patient that was unconscious and I was a brain surgeon, and I took a fine electrode and stimulated a region here, I would create a sensation in that person's knee or in that person's lip. Either a tingling or pain or pressure, depending on which particular group of neurons I had activated. Okay? So when I do light touch, it goes to the same part of the brain every single time. If I do pain, it's going to go to a different area. Now, one thing I'm going to bring out tomorrow is the thalamus does process pain. So if these were not pain, if these were not light touch or pressure neurons, but they were carrying pain, and I was deeply unconscious, not just sleeping, all right, but deeply unconscious, the thalamus could still process that as pain, but I wouldn't be able to project it. So if you pushed a pin into my hand when I'm unconscious, I'm not going to pull my hand away. Because I don't know, quote unquote, that it's my hand that's hurting. All I know is something hurts. And I'm not even thinking hurt. My body is just reacting to the pain, and the whole body moves. Because it's not projecting it back to the pain. It's feeling pain, but it doesn't know where from the body that pain is being directed, and so it doesn't project it back to any location. The whole body just tries to move to get away from it. So the thalamus will process pain. That's the only sensory aspect, but it can't do this projection. All right, so that's the primary sensory area. So aesthetic means sensory. And then obviously we have association. So you can hear something, you can feel something and know that it's smooth. You can feel it and know it's an orange versus a baseball by weight and texture. So all that's coming from association. And also there's a general association here that involves sound and vision that occurs. And that's what that cortical neglect is that I mentioned under association area. My husband has had several jobs downtown. He used to be deputy state treasurer, so he worked in the treasurer's office, and he worked for an accounting firm, so he was on 555 Capitol Mall, and he worked in a variety of components there. So I've gone and picked him up several times. So On the right side, there's a well, if I were to face with the bridge to my back, Rayleigh's Field to my back, and face the Capitol, on the right side would be Wells Fargo Bank and anyway, one of the financial, not the, you guys probably don't know what they are, it's messed my mind. Well, on the left side would be the kitchen restaurant and other buildings. And I could describe, okay, if you stood me there, I may not have the memory of it right now, but if you stood me in the middle of the mall, I could tell you, okay, I see this building, this building, this building, this statue, the waterfall, and so on. This side is that weird red statue, and so on. Person with cortical neglect doesn't have a problem with seeing. They're not blind, but if you said, tell me what you see, they would only tell you from one side. They would completely ignore the other side where their somatic area was damaged. Not because they couldn't see it, but their brain isn't acknowledging it. So the primary visual center is working, all right, but they're not processing that image and associating it, making a conscious acknowledgement of it. If you were to give them a piece of paper and put some marks on it, say, now I want you to take your pen and make crosses out of all these marks. They would say, sure, happy to do that for you. Okay, I'm done. And you'd say, well, if you look at these over here, you haven't marked through those. Could you mark through those for me as well? And they'd say, okay. And they completely ignore those. Not that their primary visual center isn't seeing them, but the rest of their brain isn't acknowledging it. If you flipped it around, turn the paper around, they would then mark over on that side only. When I was teaching at the medical school for neuroanatomy, we had a couple of neurologists from Benicia, really fun guys that would come in and talk about this stuff to the students and act out patients that they'd had with these problems. And so one would be the patient, one would be the doctor. So this one guy, he pulled his shirt out of his pants and messed up his hair and was kind of slouchy around and sat in the chair, and the doctor came over to talk to him, and he just, and the guy kept moving around, and he just wouldn't ignore him at all until the guy came on this side of him, and then he was very friendly and interacted with the doctor and answered all of his questions. All right, so that's that kind of cortical neglect. There is a very famous neurologist by the name of Oliver Sacks, S-A-C-H-S, who's written several books about his patients without giving their personal information. And one of the stories he told was of a music professor whose wife had brought him in to see him because he was the essence of an absent-minded professor, you know, bumping into the lamppost and saying, "Excuse me, sir." He wasn't recognizing people that he would meet. He seemed to know the composers whose photographs lined the main staircase in their house, but individuals colleagues he wouldn't recognize. And so they came to see the neurologist, and took off his hat and coat, and the doctor gave him a physical exam and asked him questions, and they said, okay, why don't you get your jacket back on, your shirt and jacket, and bring your hat, and we'll go out to my office. And so, excuse me, Jasmine, but the husband went over to his wife and tried to take her head off of her. And he knew he had to get something to do with the head, and he thought her head was his hat, which was hanging over on the wall. He could see it perfectly well, but he couldn't make the proper association that it was her head and not his hat. That kind of, connection of function and so on. And so that's a component of cortical neglect as well. So that often involves association areas of all of these.
How does that happen? Is it a disease that kills that side?
It's usually a viral lesion. It can be the loss of blood. So a blockage of blood supply, like a small stroke in that area. At the bottom of the primary somatic area, we have taste. Smell tends to be mostly on the bottom of that, but inside the temporal lateral sulcus. Another illustration of association with smell. When I was in graduate school, I worked weekends at Sutter Davis Hospital. and I would work 3 to 11 on Saturday and Sundays. And occasionally it was my turn to count the crash cart. And the crash cart is what you take into someone's room that's having a heart attack or some type of emergency. And it has medications, and so you have to go through and count all the medications. And it also has intubation tubes of various sizes for children through adults. And we kept these packets of intubation tubes in a box. And so I'd flip through them and say, okay, we have 3 number 8s and 5 number 7s and so on. Well, one night, I'm kneeling on the floor and I'm counting out, telling the other nurse how many there are, and I smelled mushrooms. Now, mushrooms is not an obvious hospital smell. Any of those that have been into a hospital. But I remember when I was a kid, I had gotten badges, Girl Scouts and Boy Scouts. Well, in our church, we call them Pathfinders, and boys and girls mixed together. And you answer questions and make collections and identify them, and you can get a badge in ferns or foreign languages or whatever. So to get my badge in fungi, mushroom and fungi, I had to collect a certain number and identify them, and I stored the little tree fungi in envelopes in a shoebox. And so flipping through these envelopes somehow triggered that memory of the mushroom smell of where I had stored those mushrooms from 18 years earlier, all right? So that's how our brain actually functions, because if all we had was just the auditory areas, we wouldn't be able to use the information that we get. So one of the association areas for sensory is here. It uses both parietal and temporal lobes and involves sensory speech association. It's known as Wernicke's, or more modern, the sensory speech area. And this is kind of weird. It is, your ears work fine, all right? So, clack, clack, donk, donk, goodbye. Does that make any sense to you? It didn't to me, because I just made it up, all right? But if you had damage to the sensory area for speech, when someone talked to you, that's what it would seem like. You could hear it fine, but it would make no sense to you. So nothing wrong with if they wrote the word out, you would see the word, you could see the word Carol, but it would mean nothing. It would be like it was in Arabic or Burmese, or not even or Japanese, if you don't know those. It doesn't even look like a recognizable letter. Exactly. But at least in Italian and English, you can see the words, so writing the word out doesn't help. Signing the word out doesn't help. It's the comprehension of the meaning of the word that they have lost. All right, that can be complete or it can be partial, depending on how much of that area is damaged. And that can be a real struggle to work with because we tend to be use more of one side of the brain than the other for language. If you're very young, that can be transferred to the other side. But if you're elderly, probably not. It's really difficult to relearn that. So that's a combination. And it's language, it's not sound. Okay, it's language. So some other form of language, of communication, even the meaning of music, something that might make you cry when you heard music. It's just sounds. It doesn't even have a melody anymore. Involved with this sensory area right here. Okay, we're almost done. And then we'll be done with lecture for today. So, frontal lobe, all of these have been sensory so far, right? Frontal lobe is motor, logic, and personality. I'm going to leave this diagram up here. So if you want to redraw it, because you can't erase your things. This is now going to be a section towards the front of the brain, which is going to be in the pre-central gyrus. So what I'm erasing was the post-central gyrus, and this is going to be the pre-central gyrus. We still see a little bit of the third ventricle, and we actually still see part of the thalamus. And now this is our pre-central gyrus, not the post-central gyrus. It's just anterior to the central focus. So it's this area of dark green right here. So when I talked about short associations, I talked about the premotor area. So if I decide that I am going to play Fer-de-Lis on the piano, all right, I sit down and I consciously decide where I'm going to place my fingers. And there's another piece by Beethoven that I used to have memorized, and now I only know the first 5 measures or so. Our piano is in a kind of alcove off the dining room that I walk by to my bedroom every time. And the kids are kind of disgusted because I stop and I play 5 measures, and it's 5 measures over and over because I never go any farther. But I don't have to think about what my fingers are doing. That's because short association fibers from the premotor area are going to the area where my fingers are in the precentral cortex. And so these neurons, by moving my fingers, are going to have their cell body here. And like we drew earlier, one single neuron is going to come down here, cross over in the brainstem, and then come into the spinal cord to the level of my hand, my upper extremity, and go out to those muscles that are moving my fingers. Okay? Actually, let me do this in a different color since everything up there is black. So remember, this is a projection neuron. So the cell bodies of all these motor neurons form that precentral gyrus. These neurons are identified in our pathway that we'll talk about next week as upper motor neurons. And the neurons, that's part of the central nervous system, the neurons from the spinal cord to my muscle are lower motor neurons. If I cut either one, I'm going to be paralyzed, right? So paralysis is the inability of voluntary movement. So in your lecture notes, I actually printed the wrong word, which is why I'm talking about this, because it talks about if the upper motor neuron is damaged, flaccid paralysis results. Do you see that under primary motor area on page 66? At the very bottom of that box, this damage to this region results in flaccid paralysis. Cross out the word flaccid because the term is actually spastic. So if I damage the upper motor neuron, that would be this one, anywhere along this path, spinal cord or precentral gyrus, is going to result in spastic paralysis. So again, paralysis means I cannot voluntarily move that muscle. So let's say that I was in a bicycle crash and severed my spinal cord, and I'm in the doctor's office. I can't move my legs, okay? And the doctor comes along and says, I want to test your reflexes. And he takes a reflex hammer and hits my patellar tendon. Is my leg going to move or not?
Yes.
Yes.
It will. If he tells me now, move your leg out, will I be able to do that?
No.
No. Okay? Because my upper motor neuron, where I consciously take a thought to extend my knee, there's no connection there. So why do I move my knee when he taps it? Why do I extend my knee when he taps my leg with the reflex hammer? It's a reflex. So when he taps my knee, sensory information comes into the spinal cord, and that's what activates that lower motor neuron. Okay? Not my conscious thought. So movement is still available, but not with me controlling it. So that's why we use the term spastic. Okay? The word flaccid actually applies to a lower motor neuron. So if I have polio, or my lower motor neuron is cut, and my upper motor neuron is intact, no matter how much information I send down there, it's never going to get to my muscle. the sensory information coming back in a reflex will never get to that muscle. It's never going to move. Hence, flaccid. All right, so the word flaccid in your lecture notes should be changed to spastic when it applies to upper motor.
So flaccid actually was just cut off. There's no connection.
The lower motor, yeah. There's no neural connection to the muscle at all. All right, now we talked about the premotor area, just for regular movement. We have a premotor area for speech, and that's down here, slightly separate from the precentral gyrus, and that's called the Broca's area or the motor speech area. And it's slightly similar to the sensory speech area in that I'm not paralyzed. If I have damage from a stroke to my motor speech area, I can still stick my tongue out. I can still move my lips, wah, 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 all right? But when I go to say a word, I cannot activate those muscles in the sequence required to say the word I want to say. A different word comes out or nonsense comes out. So this morning, I started to write spastic up here, and I wrote flaccid instead. My premotor area was already thinking about what I was going to write down here. And it gave me the word flaccid. My muscles made the movement for flaccid. So that's kind of what happens with Broca's area. Okay, the muscles aren't paralyzed for speech. They just aren't getting the signals to cause them to act in the right movement at the time. So you either say the wrong word or you say the same got. When I want to say cat, but coming out as got, and I hear that, and I know it's wrong, so I try to say cat, but got comes out again. And it can be very frustrating for these individuals because their sensory speech areas usually are working, and they can hear that what they're saying is not the right sound. Again, it doesn't help to write it out. They will write the wrong letters, all right? Or they will write something that doesn't look like a letter, depending on how severe this is. They can't sign because their fingers and hands will make the wrong movements. So it's any form of communication. Again, it involves music. If they try to play music, it doesn't always work. Again, depending on how severe the damage is. Okay, and then finally the prefrontal cortex and basal forebrain. So if I can borrow, then we'll be, well, one more structure and then we'll be done. So here, this inferior, using my cerebellum and my balance again. So this region right here is basal forebrain. It's what rests on the frontal bone on either side of the crista galla. This is involved in long-term memory. So you learn something, you learn a telephone number, that involves the hippocampus. But you use it over and over and over and over and over. Or your street address, and 25 years from now, even though you haven't seen that house, you still remember the street address. Then that's long-term memory, and that involves basal forebrain. Frontal cortex is this area right here. All right, basal's underneath, prefrontal cortex is right up here. And that is logical thinking. That is doing math, making decisions. Okay, that's the cognitive function. So when my son had his concussion last week, and they asked him to, gave him 5 words and asked him to recite them. That was short-term memory, all right? But then they asked him to do some other cognitive functions, do some math and so on. And that's testing this area right here. So, but that can be damaged by gunshot wounds, nail guns, and you can also see personality changes. So there may not be a loss of cognitive function, but a person develops anger issues. The case a couple of weeks ago where the woman from New Jersey or whatever was trying to ram the security gates at the White House. In her history, said she had a 1-year-old daughter, and they said while she was pregnant she had fallen down the stairs and suffered traumatic brain injury. And her employer said she had a volatile temper. She had personality changes. And so that involves frontal lobe areas.
Is that the same that people can get brain damage and it becomes really reckless.
Yeah, some of that involves limbic area that we'll talk about tomorrow as well. Okay, the last area that I want to talk about is the insula. The insula is a cortical area. It's a really weird area for what it does, but you'll know that. I think I should just take it off before I pick it up. So if I pull this part of the brain out, this is my lateral sulcus. So if I pull that open, pull this up, notice how you can see more cortical tissue inside there? All right, so this right here, that's really brain surface. It's really the cortical surface, but the parietal and temporal lobes have grown so fast that they kind of grew over it. So that's the region identified as the insula. And that's the last thing we're going to talk about. It's this region right here. So if we look at the cross section, the coronal section, here's the lateral sulcus. This is the insula here. So it's covered over by both parietal and temporal lobes. And let me get to it through here, right here. So I've cut it away. And look at what this thing does. So if they scan it by heat or follow blood flow, this area of the brain lights up when you anticipate pain, when you decide whether you want dark chocolate or milk chocolate, when you taste food and decide whether the milk is spoiled or not, when you see someone cheat and think about whether you're going to go tell the teacher, when you think about whether you're going to cheat or not. So it's these emotional decisions.
Can we have the insula damage?
Yeah, it's more likely to damage unless it's a blood supply. If you have physical damage to the outside, you're more like an epidural hematoma or a subdural hematoma. It's more likely to do the surface of the temporal lobe. So a tumor or lack of blood supply from an aneurysm is more likely to do just the insula. Okay? So it's just this weird seeing some discussed on, so it's an emotional response. You're talking to somebody and they're like, yuck, and you feel like you're being put down. So those kinds of emotional responses will affect the
I
insula.
watched a video about actors, how they actually, like, an actual acting, how it makes you feel the emotion that they're feeling is actually something that they're playing with in your brain. Is that what that is?
That is part of this, and also the cingulate and the amygdala that we'll talk about tomorrow. Okay? All right, so we're done with cerebral hemispheres. Tomorrow we're going to talk about gray matter, the basal nuclei, thalamus, and hypothalamus. Okay? And then we'll move to brainstem on Tuesday. So let's look at, I'll check with you at 1. Kind of what I did with my morning class. They weren't ready at 1. Well, they weren't ready in 45 minutes, so they took about 60 to 65 minutes or so of study time for the quiz. So I'll check with you at 1, and if you're not ready, we'll go a little bit longer, because we have even longer. You guys have it till 2 for the class period. So if you want longer for before the exam, that's fine. Just a word of caution. We don't have a lab exam on this until the 19th of November. So there is time to study, but we keep adding new material. You're not going to want to wait until November to start learning this material. And this week not this next week, but the week after, Jeff is out of town, so there won't be any open lab for that time, so just be aware of that in your use and scheduling of lab. An open lab, there is open lab next week and tomorrow. So use that material to kind of go over what we've talked about today, and especially next week. Your quiz for today, as well as the lecture quiz, does not include the Final Four. All right, so I took that off of the online lecture quiz, and it's not going to be included in today's material anyway, because today's material is what we're covering until Tuesday. So it's the neuron. All right, so the structures of the neuron, diagrams of neuroglial cells, like an astrocyte versus an oligodendrocyte, the artery, the blood supply, the folds of dura mater, and the sinuses. what are cohesive bonds? The sinuses in here, the part of the neuron, like the
Дураметр, который
dendrite.
Фокс да, все, которые Фокс Created, все, которые Фокс Created. Уже капец